Hello dear colleagues and dear guests. I'm Hakan ESKİZENGİN. I am PhD student. I'm a lecturer in Ankara University in biology department. In this presentation, I'm going to talk about the histochemical effects of spinosad on rat liver. The main purpose of research is to inform about these bioinsecticide effects. Spinosad is a bioinsecticide. This is increasingly used to neutralize harmful insects. This insecticide affects both target organisms and unfortunately affects also non-target organisms, including humans and other vertebrates. While this drug neutralizes insects, people also exposed to this substance by accident. For this reason, the effect of spinosad on the liver structure, by the way, liver is the detoxification organ of the people, was examined in this study. The liver is the body's largest gland connected to digestive tract, and this is the largest organ after skin. It weighs approximately 1.5 kilograms. The shape of the nuclei of hepatocytes is spherical or ovoid. Their size varies from cell to cell. These differences are related to the state of polyploidy. Rarely, cells with two nuclei can be seen in the liver. Pesticides are compounds used to prevent, reduce, or control harmful organisms on animals and plants. Insecticides, which are included in this group of compounds and used against insects, are also called agricultural pesticides in many places. On the other hand, they are used at home and in many other areas. In parallel with the common usage areas, insecticide poisoning is encountered in the society due to reasons such as carelessness, not using protective clothing and masks, packaging and storage mistakes, unconscious consumption, and intake for suicide purposes. Spinosad is a secondary metabolite formed as a result of aerobic fermentation of the naturally occurring soil actinomycet Saccharopolyspora spinosa. Spinosad is a potent neurotoxin against certain arthropods, particularly butterflies, beetles, and flies. Although spinosad has been identified as an insecticide with a new mode of activity, it is recognized that resistance management is necessary to maintain the long-term effectiveness of this bioinsecticide. Moreover, spinosad is considered to be a selective pesticide as it exerts little or no effect on many beneficial insects. It also exhibits little toxicity to mammals and has a favorable environmental profile as it degrades in the environment. Although there was no acute toxicity to terrestrial birds or aquatic invertebrates, spinosad-mediated cellular vacuolization has been observed in the many tissues, including the adrenal gland, liver, lymphoid cells, kidney, thyroid, stomach, and lung. Stubbins et al. Show that spinosad-mediated cellular vacuolization is resulting from the accumulation of the polar lipids in lithiums. For this research, we purchased animals from Ankara University Experimental Animals and Research Laboratory. The experiment was started after a 7-day quarantine period. Rats were divided into 14 groups. 2 of them were control groups. In our experiments, olive oil and vitamin E, which are in the second control group, were given together. The reason for this is the molecular and cellular effects of vitamin E, scavenging reactive oxygen species and nitric oxide as an antioxidant, as well as protecting cellular structures. We use hematoxylin and eosin staining technique and double staining technique for the experiments. In this research, we examine the tissue integrity, the morphological structure of the nucleus, the size of the nucleus, preservation of the integrity of the cytoplasm, and closeness of the cells. When we look at the first control group, hepatocytes are located at the homogeneous intervals, and also nuclei in almost all hepatocytes are of equal size. In the second control group, it was observed that the liver cells were radially located, and in the fluorescence microscopy images, observed that hepatocyte nuclei were in normal structure. In the third group, which was administered 9 mg spinosad for 24 hours, had a similar structure to the control group, but Kupffer cells were rarely encountered in the central vein and distal spaces. In the other group, 72 hours after administration of 9 mg spinosad showed us loss of cytoplasm in some hepatocytes. And also, we observed that the nuclear structure of some hepatocytes was disrupted and shrank. We thought that the cause of nuclear shrinkage in hepatocytes might be chromatin reduction due to the toxic effect of spinosad and necrosis of the cell. 1 week after 9 mg administration, we observed that the decrease in the nuclear size of some hepatocytes, it suggested that the cell had undergone necrosis due to spinosad. In the sixth experiment group, we also administered vitamin E. Loss of cytoplasmic material was observed in some hepatocytes, and also nuclei of some hepatocytes were observed to shrink and chromatin agglomeration was observed. 72 hours after administration, we observed a large number of erythrocytes in the central vein, and also increased number of binucleated hepatocytes and Kupffer cells were found. And also, you can see in Doppler staining, a large number of erythrocytes were detected in the central vein. 1 week after administration, we can see that cells were separated from each other, and we can see changes in nuclear size. And also, chromatin aggregation in the nuclei of some cells and Kupffer cells in distal spaces were detected under fluorescence microscopy. When we look at the 37 mg of spinosad group, the nuclei of the cells were smaller and double nuclei were found in most hepatocytes. In this group, when we look at the Doppler staining, we can see the shrinkage of the nuclei of some cells and separation of hepatocytes from each other. And also, a large number of erythrocytes were also found in distal spaces. 1 week after administration, we can see lysis of the cytoplasm material in the hepatocytes. And also, we can see an increased number of erythrocytes in the distal space and in the central vein. In this group, we observed that hepatocytes did not undergo cytoplasmic loss because of the vitamin E. Although there is vitamin E in this group, we can see that there is a loss of cytoplasm. And also, Kupffer cells were found in the distal spaces. And when we look at our final group, we can observe that there is morphological disorders, such as loss of cytoplasm, loss of chromatin in the nuclei, and fragmentation of hepatocytes. As a result of this study, we observed that 9 mg and 37.38 mg doses of spinosad had a damaging effect on the rat liver structure. In conclusion, this study showed that these two different doses of spinosad had a detrimental effect on the liver of rats after 72 hours. Biochemical effects of such pesticides have been reported in many articles. However, there are few studies on the effects on the structure of the organs. This study showed that spinosad damaged the liver structure. It is thought that the doses we apply in the fight against pesticides will be taken into account and will be a source for studies in this subject. Thank you for listening. Have a great weekend.